Hello everyone and welcome to this video on Western blot. Today we answer the question, what is a Western blot and how does the method actually work? But before we start with this video, please note that you can download these slides as well as a PDF summary explaining the whole method in detail if you support me on Patreon. You can find the link in the description below. Very often when you read a scientific article, you can come across a figure like this. This is a finished Western blot where every dark band represents a specific protein. These Western blots are used in biochemistry, in the hospital for disease tests, and as doping tests. But what is a Western blot? The Western blot is a biochemistry method which is used to detect a protein or multiple proteins of interest from a sample. So these samples can contain many things, can be a whole cell lysate, but we just want to detect one specific protein. Is it there and how much is it there? The Western blot method takes 3 steps: gel electrophoresis followed by transfer and labeling. But before we get to these 3 steps, it's very important that your samples are well prepared. For Western blots, you can use samples like whole tissues, cell culture, or blood samples. These samples all need to be lysed in order to obtain a protein lysate. It's very important that you keep these samples on ice or that you use protease inhibitors in order to prevent degradation of your proteins. In this lysate, the proteins will still have their original structures, and these need to be denatured in order to continue with the Western blot methods. You can use reducing agents or heat to denature your proteins. Once the samples are well prepared, you can move over to the Western blot method itself. As mentioned before, this method takes 3 steps: gel electrophoresis, followed by transfer and labeling. In the first step, gel electrophoresis, we will load our samples onto a polyacrylamide gel. This gel provides a thick mesh for your proteins to migrate through. Once we put our samples on top of this gel, electricity is applied and the proteins will migrate throughout the gel. Small proteins will migrate faster and big proteins will migrate slower. Once finished, you will have a gel like this. Usually, in the first lane, we will provide a standard which can be used to determine the size of the other proteins in the gel. Once gel for electrophoresis is finished, you can move over to the next step, which is transfer. In this step, we want to transfer all the proteins from the weak gel of the gel electrophoresis to a stronger membrane which can be used to detect the proteins. Again, we use electricity to our advantage to pull the negatively charged proteins out of the gel onto the membrane. On both ends a buffer is applied, and that's why this is also sometimes referred to as a sandwich. After transfer, all the proteins will be fixed to this stronger membrane and we can move over to labeling. In labeling, the goal is to visualize our proteins of interest which are fixed to the membrane. The proteins themselves are not visible and therefore we have to label them with something we can see. Here we use 2 antibodies, a primary antibody which has been specifically made to target the protein of interest, and we use a secondary antibody which is designed to target the primary antibody. The secondary antibody usually carries something that we can visualize, and that's why we can end up with a Western blot like this. This is the finished product where you can see the membrane that we have labeled with our antibodies, and we can visualize a dark band where our protein is present. We also can visualize the standard which we can use to determine the size of this protein of interest. So this is a finished Western blot. We have detected our protein and the method is finished. So to summarize, the Western blot is a biochemistry method which is used to detect a protein or multiple proteins of interest from a sample. The sample preparation is very important for the Western blot method to work. And the full Western blot method takes 3 steps: shell electrophoresis, transfer and labeling. If you like this video, please consider giving me a like and subscribing to my channel. And if you want to download these slides or the full PDF summary, you can download these if you support me on Patreon. Thank you for watching.